Hello, my name is Vincent D. Arrogante, MS Biology student. I'm going to present the synthesis report that I choose, which entitled Evolutionary Systematics of the Family Pontederiaceae. So first of all, I choose these taxa family Pontederiaceae as a preparation for my masteral thesis. I'm going to introduce first what my topic is all about and the objectives and search strategy of my synthesis report as well as the discussion and conclusion. Family Pontederiaceae is a small aquatic monocot family placed in the clade of Communales. So this clade can be morphologically characterized by its distinctly alternate bifacial leaves, perianth petaloid, then the flowers bisexual, zygomorphic, and enantiostylus. So under the Pontederiaceae, some of the examples is the water hyacinth or Echinoria craspis that is a free-floating aquatic plant. So as you can see, I provided some of the pictures that I took in 2021 at Tubaran Sur, Danao, Cebu. The main focus of the synthesis report is to know the evolutionary and phylogenetic systematics of the family Pontederiaceae. So this report specifically aims the following. First is to know its morphological phylogenies with evolutionary perspective, to review its morphological and molecular data, then to gather data about its molecular clock, then to research its evolutionary systematics on biodiversity conservation, and lastly is to understand its climate change research. In order to find articles about the morphological and molecular phylogeny of the family Pontederiaceae, I searched several online websites such as PubMed Database, Frontiers, Google Scholar, Science Direct, and other reliable sources with a search period of 1973 to 2022. For the discussion, when talking about its morphological phylogenies with evolutionary perspective, the Pontederiaceae are distinctive in being emergent to free-floating aquatic herbs with simple, sheathing bifacial leaves, then actinomorphic or zygomorphic flowers, and lastly, di or trisulculate pollens. The phylogenetic tree represents the relationship between the monocotyledon in Figure 2 and phylogenetic tree of Conmenelalis clade, highlighting the family Pontederiaceae. Then the family is arranged in four genera, which are the Echinoria, Heterantera, Monocoria, and the Pontederia. For morphological analysis, according to Pellegrini et al. 2018, the Pontederiaceae and the Hemodoraceae clade is supported by seven characters, which are the presence of the septal nectaries, perianth 6-lobe, perianth with 3 plus 3 arrangement, epithelialous stamens, then stamens dimorphic, then endothecium with a basal thickening, and lastly the non-tectate columnate exine. The Pontederiaceae are monophyletic with high statistical support such as the dimorphic leaves, then leaf blades late bifacial, then perianth ranging from lilac to purple or blue. Then lastly, the posterior lobe um, with a nectar guide and also the presence of the antocarp. While the Hemodoraceae supported by plants, mostly spirally alternate sessile leaves, basal bract, conduplicate, main florence reduced to a solitary cincinus, then sparse erythematous tissues in the perianth and unevenly trilobate stigma. For morphology and its systematics, the evolutionary relevance of the bifacial leaves is significantly harder to infer, since unifacial leaves are noticeably common in several aquatic plants. The reversal from equitant to alternate leaves seems to be a byproduct from the reversion from unifacial to bifacial leaves. The involate six in Potenderaceae extremely unusual since the blade of the new leaf encloses the petiole of the preceding leaf. This feature is also unique in the angiosperm and is easily observed in most species in the family but is especially obvious in Echinoria crassipes. This feature might also be related to the adaptive shift and radiation to a complete aquatic lifestyle in Pontederiaceae, being most likely a result of the reversion to bifacial leaves. Then, development studies focusing on the ontology of the leaves in Pontederiaceae in comparison to some of the um, Hemodoraceae and Philidraceae might help us better understand the mechanics of the reversal from unifacial to bifacial leaves in family and how this shift might have affected general leaf morphology and also the appearance of the novel structures such as the ligule. Besides the morphological data and phylogenies, I also provided some of the molecular data of Pontederiaceae. So the topologies recovered from the combined plastids and the total evidence database strongly corroborate the bigeneric circumscription of Pontederiaceae. They are also congruent with these previous phylogenetic studies using the molecular and combined data sets, and also the partially congruent with the morphological based phylogenetic tree of Pellegrini et al. 2018. So most molecular studies in the family recover a well-supported Pontederiaceae divided into two main lineage, so corresponding to a well-supported Heterantera and poorly supported Pontederia, which highlighted in the red box in this figure. So using the NDHF, then the RBCL, plus a restriction site in the chloroplast genome by Graham et al. 1998, and five nuclear gene families recovered employing in an express sequence tag or the EST. So as in previous studies by Graham, they recover Pontederia arranged in five main lineage, each representing a well-supported morphologically group such as the Echinoria, Paniculata group, then the Monocoria, then Echinoria crassipes group, Echinoria, and then lastly the Pontederia. So the monophily of the Heterantera is indisputable, and the inclusion of hydrotrix and the scleropsis in Heterantera was strongly corroborated. The congruence between the results of Chase et al. 2006 lends supports to the idea that the molecular data have now converged to the correct high order relationship within this order. For molecular clock, an alternative approach to assess the importance of an outgroup in routing the tree is explored by Graham et al. 2002. So this is by establishing whether the outgroup provides a sufficient signal in response to root location, indicative of historical linkage, or due to long branch attraction, or also called as the LBA. So using the Pontederiaceae, an aquatic monocot as the case study, they assess how the nearest outgroup provides for routing the Pontederiaceae compared to those less closely related relatives and further investigate the role of the LBA when determining the optimal routing of Pontederiaceae. However, they conducted that LBA may influence routing and may be supporting the wrong outgroup. Then, to further reduce the LBA and to ensure robustness of the outgroup routing method, they recommend to their studies multiple sampling of outgroups within the sister group rather than sampling within less closely related taxa. So, as you can see in Figure 5, bootstrap 50% consensus trees of phylogenetic analysis of monocot relationship um, is seen in this figure. So, in A, modified from Chase et al. 2000, based on plastid ATBP and RBC L, and nuclear 18S rDNA, and B, modified based on the plastid ATBP, then the RBCL, then the MATK, and NDHF. Then also mitochondrial ATPA and also the nuclear 18S and 26S rDNA. So number of asterisks indicates general range of bootstrap percentage. Then names of the orders follows the APG2 in 2003 except for that Petrosaviales, and the size of the triangle indicates roughly the size of the clades in terms of the species. Due to its rapid rate of growth and reproduction, this weed can compete other aquatic plants. Then the large, dense floating mats of water hyacinths blocks the sunlight and monopolies nutrients, killing submerged aquatic plants. Then the mats not only blocks the sunlight and preventing phytoplankton and submerged plants from photosynthesizing, they also prevent oxygen from dissolving into the water, thus causing an overall drop in dissolved oxygen concentrations. Then the aquatic organisms sensitive to changes in dissolved oxygen levels will have a negatively affected regarding their biodiversity conservation. Then, however, the complex root systems and the leaves of water hyacinth mats may also provide refuge for aquatic organisms, especially epiphytic invertebrates like snails and aquatic insects. So, population composition can also be affected as well. So, in some areas, fish populations have an increase due to the water hyacinth. So the overall effect of the water hyacinth invasions on biodiversity is dependent on many factors. So as you can see in figure 6, it shows interactions between water hyacinth and other components of the freshwater community. So the introduction of water hyacinth has an effect uh, to a freshwater ecosystem through its interaction with all of this part of the ecosystem, which interact with each other as well as making it difficult to predict the exact ecological effects of an invasion. So also in some other countries like Kenya, water hyacinth was perceived to affect fisheries through reduced levels of production, then a reduction in species diversity, then poor quality fishing, then rising costs of the operation resulting in lower incomes to fishers and higher prices to consumers. Based on some articles, how water hyacinths will survive to climate change. So scientists in Hubei, Southeast China set up greenhouse experiment to examine the effects of the temperature on the survival and growth of water hyacinth. The experiments ran for 3 months and plant survival was significantly affected by both temperature and the size of the stem. All floating plants with large stems survive, but in those with small stems, survival increased with temperature because the stem base is where carbohydrates are stored as energy remains are reserved in temperature. So a larger stem improves the chance of overwintering survival. So in the case of the rooted plants, which were kept in moist sediments without surface water, survival was much more higher when the stem base was buried in the sediments and insulated from the cooler air. So as with survival, plant growth increased with temperature and with the size of the stem base. So as temperature increases, relatively more biomass was allocated to shoots and less to stems. So therefore, it seems that the rising of the winter or the temperatures will have a strong positive effect on the survival and growth of water hyacinths because of its ability to shade out competing aquatic plants and also its ability to capacity to expand its geographical distribution. For the conclusion, Pontederiaceae is one of the first family of flowering plants to be focused of studies dealing with its phylogenetic history based on morphological, molecular, and combined data. Nonetheless, until very recently, the taxonomy of the family remained dogmatic and outdated with the recognition of several non-monophyletic taxa. The arguments used as a basis for maintaining such assemblages are based especially on misunderstanding of the principles of phylogenetic systematics. One of the main paradigms of modern phylogenetic systematics is the proposal of classification system that accurately reflect the evolutionary history of the studied group, being simultaneously easy to use. So in order to achieve that, novel classification system should be based on the molecular phylogenetic studies together with morphological and whenever possible, also including the less common characters such as the anatomy, ecology, geography, then the phytochemistry. Furthermore, without the inclusion of the morphological characters in phylogenetic analysis, there is no way to obtain the morphological synapomorphism to support the recovery relationship and any proposed new classification. So the implementation of these ideas on the systematics of Pontederiaceae has generated not only to the monophyletic genera but also to considerably facilitated the taxonomy of the group. So with the classification implemented, species of the Pontederiaceae are easily and ambiguously placed under the two genera supported by morphological and molecular data. So an infrafamiliar classification for the Pontederiaceae has always been a little taxonomic and systematic relevance due to the family's reduced size. Then, with Pontederiaceae consisting now of only two genera, the recognition of subfamilies and tribes seems rather pointless since each genus would be placed in its own subfamily. So, it is conceivable to integrate the two types of data, which are the morphological and molecular, and doing so occasionally leads to synergistic improvements in support. So, Echinoria crassipes or water hyacinths, a member of the Pontederaceae, competes other water plants due to its quick pace of growth and reproduction. So the massive thick floating carpets monopolize the resources and blocks the sunlight and destroys submerged aquatic vegetation. So overall, this synthesis report talks about the how useful the morphology and the molecular data in integrating a or creating a phylogeny. So thank you. That would be all.